Okay, guys, we're back. So talking about lateral inhibition, we've already said what lateral inhibition is. It literally is inhibition coming from the sides. So it's inhibitory signals converging onto a cell in the retina that is coming from the sides. This actually explains this illusion called the Hermann grid, this illusion where you see in your periphery, in your peripheral vision, these little gray blobs at the intersections of all of these white lines. So let's take a look and see exactly what is going on here and how lateral inhibition explains this phenomenon or this illusion. So here on the left, under above A here, is the Hermann grid, right? So this is just sort of one block of it, or one piece of it, showing one set of intersecting white lines, right? So there's a gray black square here, 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 and here, and here's those intersecting white corridors, the place where you would see a black blob right around here if this was not in the center of your focus, right? If you weren't looking directly at here. So why does this happen? We said that it's because of lateral inhibition. So let's assume or imagine what would happen if we could sort of draw out a neural circuit that explains this. So here is this same picture of the Hermann grid, but just from somewhat of a different angle. Imagine looking at it from slightly overhead and at an angle. So each of these little gray things here is one of the black squares of the Hermann grid. And each of these green things is a receptor, is a photoreceptor, a sensory neuron, either a rod or a cone in your retina. Okay? And so you'll notice that each of these receptors is located over a different portion of the Hermann grid, right, which maps onto where you would see them here above the Hermann grid, right? So let's start with receptor A. Receptor A is located right over the intersection of the two white lines, so it's right at the intersection there, right? So we could see receptor A there. Receptor B is in one of the corridors down here. Receptor C is over here. Receptor D is up here, and receptor E is over here. Okay, and so let's think about the activity of these receptors, the rate at which they are firing and generating action potentials in response to the stimulus that's out there in the environment. And what is the stimulus? It's the Hermann grid, right? So let's think about what the activity of each of these 5 receptors might look like, okay? So what we also have drawn out here is sort of showing the way in which these receptors are sending their signals vertically, right, to other cells in the retina. So each of these blue objects here is a bipolar cell in the ganglia, in the retina. Okay, so each of these receptors is synapsing onto a different bipolar cell in the retina. So what is the activity of each of these receptors look like to begin with? Well, how much is each receptor being stimulated? How intense is the stimulus that's hitting each of these receptors? Well, each of these receptors is located over a white area of the picture. So they're all being equally stimulated, right? So they all should be sending at this point signals that are all of exactly the same strength, right? So they should all, since they're all being stimulated by an equally intense, an equally bright surface, equally reflective surface, they're all located right above the white portion of the Hermann grid, they're all being equally stimulated, and they should all be generating action potentials at about the same rate. So they should all be generating, all should be firing just as rapidly as one another. So they should all be sending pretty much the same amount of excitatory input to each of their bipolar cells. Okay. And that's important because the amount of, or the activity of a cell is going to determine our perception, right? And not just the activity of a receptor, but ultimately because the receptors are just relaying their message to the bipolar cells, which will eventually relay their messages to ganglion cells down here, right? It's going to be ultimately the ganglion cells that have the final say in what we perceive, but the bipolar cells are going to be important as well. So we need to consider, at least for this example, what the activity of the bipolar cells is going to look like. Okay, so we would think that because each of these receptors, A, B, C, D, and E, is being equally stimulated, because each of them is located over a white part of the grid, they're all being equally stimulated, they're all generating action potentials at the same rate, they're all firing at the same rate, they're all relaying to their bipolar cells exactly the same degree of excitatory messages. We would think that we would perceive the same thing, but we don't. And the reason has to do with lateral inhibition. So what is lateral inhibition? Inhibition coming from the sides. So you'll notice that, as we've said before, signals in the retina don't just travel vertically from the receptor down to the ganglion cells and then out up the optic nerve. They also travel horizontally. And that is what these arrows here are showing. This is lateral inhibition. So each of these bipolar cells, these four that I have sort of colored in red, are sending inhibitory signals in to this ganglion cell that is receiving messages or signals from receptor A. And so the more these receptors, B, C, D, and E, the more they're being stimulated, guess what? The more inhibition they're going to send this way. So each of these four receptors is being stimulated pretty strongly because they're located over a very reflective surface, right? So they're generating really rapid action potentials. And because they're being very sort of, being stimulated very strongly, they're sending lots of inhibition laterally to the ganglion cell here that's situated below receptor A. And so what does that mean? If this ganglion cell, which is receiving a lot of excitatory input from receptor A, because as far as receptor A is concerned, what's out there in the environment is a white stimulus. But the receptor doesn't have the final say in what we perceive, right? The activity of the ganglion cell matters. And so what is the activity of that ganglion cell going to look like? Well, if it's receiving a lot of excitatory messages from receptor A, then it's likely to generate a lot of action potentials. But what have we said about the activity of a neuron? The activity of a neuron depends on the sum total of all of the inputs that are going to that neuron. So the activity of this cell doesn't just depend on the amount of excitatory messages coming from receptor A, also depends on all of these inhibitory inputs that are coming from all of these four other bipolar cells. And so what's happening is that all of this this inhibition coming from the side, right, this lateral inhibition, is turning down the activity of this cell and basically counteracting the excitatory messages that are being sent by receptor A. And so what does that do? It's turning off this bipolar cell. This bipolar cell is getting weaker and weaker and weaker. It's firing fewer action potentials. And so what does that mean in terms of our perception? Well, our perception of brightness depends on how rapidly neurons are firing, right? We've said that before in terms of how different characteristics of a stimulus are coded at the level of the retina. Stimulus intensity is coded by the rate at which neurons fire. So a bright light leads to a perception of something that, or very fast, rapid firing of a cell. So generating lots and lots and lots of action potentials leads to a perception that something is bright or white, whereas slower responding from a photoreceptor leads to a perception that something is dark or grayish. And so what does it mean that the ganglion cell that's right below receptor A, right below, which is, located here at the intersection of these two white lines, the ganglion cell below receptor A, even though it's getting excitatory messages from A, is being turned down through lateral inhibition. So the rate at which this ganglion cell is generating action potentials is slowing down. It's generating far fewer action potentials due to lateral inhibition. And so because this ganglion cell is firing at a slower rate now than it would be if it weren't for this lateral inhibition, that's what leads to the perception of the dark spot at the intersection of the two white lines. The fact that the ganglion cell that is situated below the receptor, that's right there at the intersection of those two white lines, that ganglion cell is essentially being turned off through lateral inhibition. Right? So this cell is not generating action potentials as rapidly. And so our brain interprets that as, well, receptor A must just be getting stimulated by a fairly weak, intense, dim light, or by something that's, in other words, kind of gray or kind of black. And so that is what leads to our perception of these little black blobs located at the intersection of these white lines. The fact that lateral inhibition turns down the firing rate of this ganglion cell. Okay, so I think that is enough for right now. That is the explanation for the Hermann grid in terms of lateral inhibition. Lateral inhibition, as your book talks about, can also explain several other phenomenon such as Mach bands and simultaneous contrast as well. So we can talk a little bit more about that in class. And certainly if there's questions about this, feel free to let me know. Otherwise, I will see you guys in class most likely on Thursday.